Yeah, we are waiting to start.
Ni Good morning, everyone. I will just give it an extra minute or two because I can see that people are still coming in. So just making sure that everyone's settled. All right. Uh, okay. People are still coming in, but we'd like to start now so that we can keep time. Good morning, everyone, and welcome to the WIO Restore Inception meeting. My name is Vera Horigue. I'm a research fellow at Macquarie University and WIOMSA and one of the leads of this project. So I will serve as the main host for this event, and it's wonderful to have all of you here today. Our colleagues from different parts of the WIO and also other parts of the globe. So before I call on our fearless leader, Dr. Arthur Tuda, to officially open this event, I would just like to remind people about some practicalities. So please keep your microphone muted unless you're presenting or invited to speak. We'll open for questions at the end, and this will be moderated by Dr. Juliet Karisa, one of the project leads. And if you have a question, please use the raise hand reaction or you can drop it in the chat. And if we don't reach your question in time at the end at the session, the Q&A session, you can type it in the chat and we'll follow up by email. So as I mentioned in the email over the weekend, which I apologize for sending, our meeting is structured in 90 minutes. So in the first hour, we'll just have a series of presentations, and at the end is the Q&A. We'll start with a general overview of the project by Dr. Arthur Tuda, and then presentations by other project leads. So distinguished Professor Peter Harrison and Dr. Dexter de la Cruz from Southern Cross University will be talking about the larval restoration science and symbiont culture work. And then Professor Gildas Todinanahary covers the arms presentation. And then I will give a talk along with Attorney Yvonne Waweru about the governance research and OGA framework. And then we have a Q&A moderated afterwards, and then the next steps in closing. So I'll just call on Dr. Arthur Tuda to formally open the meeting.
Great, Vera. Thank you. And very happy to see you all. As soon as we started the meeting, my power went off. Luckily, I can connect via phone. Thank you very much, colleagues, and it's nice to see you this morning. Some of you it's afternoon and evening probably. But just a quick opening remarks, not so long. For those who don't know who WIOMSA is the Western Indian Ocean Marine Science Association. And as Vera has introduced me, I'm the executive director, but also the project lead for this WIO, of course, with the other leads as well, who you'll hear from shortly. So at WIOMSA, what do we do? Our role mainly is to connect science, policy, and practice across the Western Indian Ocean region. For those who know the geography of the Western Indian Ocean region, we cover 10 countries, the coastal strip running from Somalia all the way to South Africa and the islands of the Indian Ocean. And over the years, we have supported a lot of activities in terms of science, policy, and management from marine protected areas, fisheries governance, and regional collaboration. Vera, you want to run this?
I'm sorry, Arthur? Oh, he froze.
Slide, this the first slide, probably.
Okay, yeah, sorry, we lost you there for a bit. Okay, moving on now.
Yeah, sorry. All right, sorry. Just the first slide. Yeah, sorry about that. We have some power issues here. It's been raining, so yeah. So I was just talking about WIOMSA and who we are, just for the purpose of participants who are here to understand what we do. But I think before I say much, in my 25 years working as a marine protected area, I mean, I've seen reefs come in different forms. So I've seen some that are thriving. We've seen recoveries in protected areas and areas outside protected areas, but we've also seen serious declines, okay? And what has changed really is that the pace of degradation is now faster than the pace of natural recovery. And what that means is that we can no longer rely on nature alone, but question we have to ask ourselves is really, how do we restore at a scale with science and in a way that lasts? So this project that we are going to present to you this morning is designed around answering that particular question. What we are responding to now is basically the pace of change that we are seeing that coral reefs cannot really cope with. And this project comes at a time when coral reefs in the WIO, as we know them, I'm happy to see some experts on coral reefs like Dr. Tima here, will tell us that they're declining at a very serious rate, and live corals have been declining for decades, and natural recovery can no longer, can no longer keep pace with this degradation, especially under climate change. So we are facing three interconnected challenges. The first, of course, is most restoration still relies on, coral gardening, which is valuable but difficult to scale and is limited in genetic diversity. The second issue we're facing, of course, is capacity. So advanced restoration science is still limited in the region. So we must build this capacity here and in our own institutions. And governance, this is the most critical, actually. Restoration today is fragmented as we see it. It's mainly project-driven, and it's weakly embedded in policy and finance. So without solving governance, even the best science will not last. Vera, the next slide, please. Yeah, so we are not dealing with the stable system, as we've said. We are dealing with a system that is under constant stress. And so our vision is to transform coral reef restoration in the WIO by combining innovative level restoration science, with strong governance frameworks, regional capacity, and ensure that restored reefs remain ecologically resilient, socially supported, and economically viable beyond the project's lifetime. So our project is 36 months, and we're going to implement it in Kenya and in Madagascar. Next slide. Great. So what is our response? Our vision is simple, as I've read, and ambitious, of course, which is to transform coral reef restoration in the WIO region by combining science, strong governance, and building regional capacity. But because success is not just about restoring reefs, it's about ensuring that reefs are restored and are ecologically resilient, socially supported, and of course, economically viable. So our approach is in 3 phases. The first is a foundational phase, which will last 1 month to 18 months. Here, we want to build capacity of the system by training partners, setting up sites, deploying level restoration, starting governance assessments. This is where the capacity is created. So the first phase will be focused a lot on building capacity. And then the second phase, we start off the monitoring and evaluating and also taking adaptive action. Here we'll track coral survival. We'll assess biodiversity, refine techniques, and also deepen governance analysis. This is where knowledge is now generated. And of course, as we head towards the project lifetime, we'll be doing policy briefs, regional frameworks, and also strengthening pathways for scaling. This is where the system becomes, we talk about sustainability. So these are not necessarily going to run as separate phases, but this is just designed to show you how the project is currently structured. I think, Vera, there's a last slide for me. Great. So, yeah, no, the one that you had before.
Okay. Yeah,
so this work is made possible through strong partnerships. Partnerships is one of the things that WIOMSA values a lot. So WIOMSA will be the lead coordinator in terms of leading the project. Also in terms of governance, but we're also working very close hands, University of Toliara, just together with the Kenya Marine and Fisheries Research Institute. At the core of this project are 4 key innovations. The level box, which is will be scaling the level deployment. The ARMS, Autonomous Reef Monitoring Structure, which will be tracking biodiversity recovery. The Symbio that is a difficult word for me to pronounce, but it's aimed at improving thermal resilience. Yeah. Whoever wrote it there will pronounce it later. And of course, ocean governance accounting. So this is an interesting one. We want to see how ocean accounting can be incorporated into coral reef restoration. So bringing in the issues of finance, but also ensuring that coral reefs are integrated into national accounting systems. So Vera, I think that is so much that I can say for the opening, and I want to hand it over to you and thank everyone who's here today and all the partners who have been involved in putting this together. Thank you very much.
Thank you, Arthur. And just to add, in terms of the project at the end, which I think Arthur has already delivered in terms of the discussion of the phases, the 3 years. But basically, this project aims to provide capacity building to at least 30 individuals across Madagascar and Kenya, and hopefully others. And then have at least 10 or around 10 plus larval restoration sites. We'd like to have an updated costing of the larval restoration method for the WIO, because there's costing about this for Australia and the Philippines, which Peter and Dexter will talk more about later. And again, create that policy document on the restoration governance framework, which we hope could be shared across the partners and colleagues in the WIO for integration and awareness. So I'll pass the presentation now to Professor Peter Harrison and Dr. Dexter de la Cruz to start talking about the larval restoration techniques. So I'll stop sharing. Peter, it's your turn.
Thank you. Hi, and thanks Vera and Arthur for that great introduction that sets the scene to the process. And thank you everyone for coming online. I can see there's 56 participants, which is great. So a great diversity of people. And this is just the start of the project. So this next presentation is very much a very short, structured presentation to give you some insights into the processes of larval restoration and setting the scene for the work we hope to do with many of you over the next few years. So I will start sharing my presentation. Okay, so this next part of the presentation is by myself and Dr. Dexter de la Cruz. We're based at Southern Cross University on the furthest eastern point of Australia. Dexter's currently in the Philippines, helping to manage our Australian government-funded ACR project with a big team of people there. And Dexter and I will be tag teaming a little bit during this next short presentation. What we want to talk to you about is larval-based coral propagation for restoration. And during this process, we use natural coral spawning events that create billions of genetically diverse larvae from a whole range of species. We generate millions of tiny microscopic larvae, and we use those larvae to restore degraded reefs. And an example here is from the team in the Philippines, 3 years after mass larval release on a formerly highly degraded site with virtually no corals. And we'll explain a little bit more about that soon. So, as we all know, reef building corals are essential for healthy reefs. And we know that reefs have great ecological, socioeconomic, and many cultural values in many reef nations around the world. As many of you would know, these extraordinary invertebrates are symbiotic habitat engineers. So the many millions of tiny microscopic algae within their tissues capture sunlight energy to create sufficient energy for these corals to calcify fast enough to build reefs. As a consequence of that extraordinary reef building ability, they create habitats for many other reef creatures, including those that are directly important culturally or economically, food security, etc., for many people in reef communities. We also know that sexual reproduction and asexual reproduction are really important as well. Reproduction is essential to all life forms, and corals have two forms of reproduction. Sexual reproduction, involving the release of eggs and sperm and genetic recombination, and asexual growth and fragmentation, particularly amongst some corals such as branching corals. Those two forms of reproduction create interesting pathways for different types of restoration. And Arthur mentioned in his introductory talk that, as you would probably be aware, most restoration projects around the world rely on asexual fragmentation and coral gardening. This can be very useful at small scale to rapidly generate new corals on degraded reefs or those that have been impacted by various events. However, in terms of scalability and cost effectiveness, all of the modeling currently shows that that form of restoration cannot be meaningfully scaled at a sufficiently low cost and generate sufficient genetic diversity to guarantee some successful outcomes at large scales in the future. And that is why increasingly around the world, many research groups are now working with sexual reproduction, which naturally increases genetic diversity, and as a consequence of that, adaptive evolution and resilience potential. So that's what we're going to talk about mostly during this presentation. We know that sexual reproduction is essential for population recovery, resilience, and adaptive evolution, particularly in response to rapidly changing climates and reef environments. We know that strong larval supply is essential for settlement, recruitment, and natural recovery. It's what keeps coral systems healthy in response to natural disturbances. However, as we're all very aware, and as Arthur has pointed out, we know that most reef systems around the world are degrading faster than natural recovery now allows. And the primary reason for this is that we're losing breeding corals, which limits larval supply and natural recovery for many of the key reef-building corals. As a consequence of that, we get negative feedback loops, which further constrain the capacity for natural regeneration and recovery. So in response to that, what we've been working on is a process called reef-based coral larval restoration, sometimes referred to as coral IVF. And what we aim to do here is use the natural sexual reproduction process of corals to increase larval supply to restore resilient breeding populations for future healthy reefs. Globally, of the more than 400 species of reef-building corals that have been studied, we know that about 85% of them are what we call broadcast spawners. So they release eggs and sperm into the sea for external fertilization. The other 15% of coral species that have been studied brood their larvae, usually through some form of internal fertilization, and the larvae are released at an advanced stage of development. The coral life cycle includes the microscopic larval phase, which settles, and the new polyp gradually grows into a series of adult polyp-based colonies. So let's look at the reproductive cycle. Inside many of these hermaphrodites, such as the branching corals, the Acropora species, over a period of about 9 months, these tiny little eggs develop. And when they get to maturity at about 9 months, there are both eggs and sperm present in each of the polyps. In many cases, the eggs become brightly pigmented, and therefore if you carefully fracture a branch of these colonies and you see these brightly coloured eggs and the large creamy or white-coloured spermaries nearby, you know that these colonies have mature eggs and sperm and will spawn during the next lunar cycle. When spawning occurs, the eggs and sperm are wrapped into bundles that are usually buoyant and float to the sea surface, and they break apart, and that's where cross-fertilization and genetic diversity occurs. The embryos develop after the first cleavage overnight and then grow up into a tiny little ball that is covered with external cilia and starts to rotate. And at that point, after about 24 to 36 hours, we call it a larva. Over the next 4 to 6 days, those larvae develop and mature. They become elongated. And during this period, many of those larvae actually are dispersed away from their parent reefs. And as a consequence of that, many of the billions of larvae that are developed naturally from mass spawning processes end up disappearing from reefs and away from target reef sites. For those larvae that do find a reef and find somewhere successful to settle, they start testing the environment, testing the hard dead coral surfaces, often with crustose coralline algae, and then they metamorphose into a polyp. After about 36 to 48 hours, the skeleton starts to form, and after a few days, symbiosis occurs so that the tiny microscopic dinoflagellate symbiodiniaceae come into the coral tissue, and that enables that tissue to start getting an extra energy supply. And that can make the difference between life and death for these juvenile polyps. At this stage, many of the initial polyps end up dying, probably because most of them end up running out of energy and some of them get eaten or scraped away by reef scrapers. For those lucky few that survive, they start growing large enough to start budding asexually, so they clone themselves to create a colony. And in the Philippines, after about 2 years from this microscopic millimeter-sized initial polyp, you can get to small plate size. And then at 3 years, they can grow to sexual reproductive size at about dinner plate size. And at that point, they then close the life cycle. The key point from all of this is that life for a larva or a juvenile coral is very tenuous. And so we estimate that possibly only 1 in a million larvae successfully settle, survive, and grow into adult corals from these spawning events. So this reproductive process works because after mass spawning events, literally billions of larvae can be generated. But the problem we face now globally is that we're losing those breeding corals that I mentioned. And as a consequence, we no longer can guarantee billions of genetically diverse larvae being produced. And that's where we start to step in to try and work out how we can use these natural sexual reproductive cycles to enhance larval restoration and recovery. So as a consequence of this process, we've come up with a reef-based larval restoration process, as I said, sometimes referred to as coral IVF. And it's designed to resupply reefs that have been degraded and damaged and those that have insufficient natural larval supply to recover quickly enough. So first of all, we go out and locate healthy breeding corals. And most of these on most reef systems now tend to be heat tolerant because those are the only corals that are left on bleaching impacted reef systems. We wait for the corals to spawn. Then we collect the spawn either in spawn cones, in scoop nets at the surface, or in passive spawn catchers that can collect hundreds of millions of eggs. We then combine the eggs and sperm from all of these different colonies to maximize fertilization and genetic diversity. And then we transfer the spawn across into these innovative inflatable larval pools. The pools that you see in this image are about 4 by 4 meters. They contain about 15 to 20 million larvae. And in recent experiments on the reef, we've been able to push that production rate up to about 50 million larvae per pool. The larvae develop over about 4 to 6 days until they're absolutely desperate to settle, and at which point we can then start to think about how we're going to release those larvae to increase the amount of settlement that's occurring on degraded reef systems. So we can either settle the larvae directly on dead corals or on limestone tiles or other forms of settlement devices directly within the pools. We can be thinking about adding heat-tolerant symbionts to the cultures to increase uptake by the larvae or the newly settled corals. We can free release these as larval clouds containing millions of larvae onto degraded reef systems, or we can constrain and direct those releases through pipes from the bottom of the net system and in innovative larval boxes. So I'll now hand over to my colleague, Dexter, and he will talk about one of the innovations that's happened in the last couple of years in the Philippines project.
Thank you, Peter. So as Peter had mentioned, there are several ways how to really release competent larvae to degraded reefs. And the release of the larvae is really important because it dictates the scale in which you can restore a degraded reef. And through the years, we tested several methods. And one of the methods that we use is what we call the coral larval box. So basically, this is a cost-effective way to release the larvae because we're working on different regions here in the Philippines, and the localities needs to adapt and create their own system to release the larvae. So basically, this larval box is made of commercially available food-grade lunch boxes, as you can see. And then we created a window with mesh on the side to keep the water flow. And at the bottom of the boxes, there are small holes. And we tested several sizes of holes, and we found out that 3 millimeter holes is it's like a sweet spot for the larvae to come out. So what we do is after the culture period, we filter the competent larvae and we uh, put them inside the box. And these boxes are placed on top of the degraded leaf. So as you can see, the bottom part of the larval box is really close to the substrate. And the larvae are slowly being released on the bottom, and it will be dispersed by natural water movement. Yeah, next, please. So this is just one of the results of our experiments here in the Philippines. As you can see in the treatment plot, there are really high settlement of larvae surrounding the larval box and zero in control. So meaning that once these larvae were released inside the box, they can settle immediately on the vicinity of the degraded reef. And on the right side in letter A, as you can see, the circular pattern is that the center of the larval box, the tiles that are close to the larval box will have the highest number of settlement. But as you go out, the number of settlers still decrease, but still we detected some settlers. So this area is around 5 meters by 5 meters. And majority of the larvae in each of the settlement tiles that we deployed, majority of the larvae settled at the bottom as expected, and some on top of the tiles and few on the side part. So it means that the larval box is really cost effective and good for settling larvae. And one of the advantages of using the larval box, because since we started the larval enhancement restoration both in the Philippines and Australia, most of our sites are in shallow areas, less than 5 meters or 6, because that's the operational limit that we can do scuba diving for longer period of time. But because of this larval box, we were able to do some restoration sites in different areas around more than 10 meters in deep. And so we developed this drop and go larval box where we just leave the larval box there with coral larvae and let the larvae disperse on their own and settle on the degraded reef. Next, please.
Thank you, Dexter. So, so as we mentioned, there's a range of different processes for effectively settling the larvae. And as a consequence of that, as Dexter's information was just showing, we then monitor settlement. And then over time in the longer term projects, we monitor the survival and growth of the resulting corals through to sexual reproduction and the closing of the life cycle. So ultimately, we're aiming not only to restore corals, we're aiming to restore genetically diverse breeding corals on degraded reefs. We've done a lot of work over the last 12 years, and we've now had more than 50 successful reef trials in the Philippines, extensively on the Great Barrier Reef, and increasingly through engaging with local key stakeholders in the Philippines, on the Great Barrier Reef, and in the Maldives, and in some other locations. And that's the foundation for what we want to do in this project with key stakeholders in the Western Indian Ocean. So some outcomes from some of these projects. Obviously, there are a lot of projects, and we're just going to highlight a few. So I'll ask Dexter again to speak to this slide, which forms part of his earlier PhD on larval restoration in the Philippines.
Yep, thank you. So when we first starting this project in the Philippines, we need to demonstrate that whether when we release the larvae, they will settle on degraded reef. So on our pilot studies, as you can see from the photo, we covered a portion of a reef using a mesh matting. So this mesh matting is again locally available and just created by our local zoomaster. And then we release around 600,000 larvae in each of that matting enclosure. And as you can see from the initial settlement that we have at least 25 settlers per tile, and that And then after 9 months, the totally barren plot where we released the larvae is now covered with corals, as you can see from the photo. So these are all corals from our reseeding experiment. And after 3 years, we saw that they started to spawn as well. And we used the spawn to restore different degraded reef areas in the same localities.
And in contrast, unlike the larval restored areas, unfortunately, the control reefs, as we said, in many of these areas, there are just too few breeding corals on many of these areas in the Philippines. And as a result of that, the control areas, the plots that didn't have larvae supplied, still look highly degraded with relatively few corals on them. And you can see the difference between the restored reef after 3, 4, or 5 years in the background there, and the control plots after 5 years, which haven't recovered. So as Dexter said, we've done a series of experiments based on this initial work, and we did another high density experiment in 2016 where we used 3 times as many larvae, partly from the first spawning of that restored population, plus larvae from wild colonies. And as a result of tripling the density of larvae onto new restoration plots, we restored high densities of the breeding population. And remarkably, we found that some of these corals were growing so quickly on these highly degraded reefs that they were breeding by 2 years, which is a world record for these types of corals. So we've restored genetically diverse breeding populations within 2 to 3 years. And when we did a cost appraisal of this, we were getting down to about $17 per adult breeding coral. That sounds quite good, but it's still too high to be cost effective for large scales. So what we've done now is tried to optimize the process to get those costs down to a more manageable level. The interesting thing from these early experiments is that we've been able to capture some of the spawn to create more larvae and then disperse those to nearby reefs, therefore increasing the scale of the outcomes from these early studies. And we've also done some extensive long-term monitoring of the fish populations. And within a couple of years, we get increased pomocentrid and butterfly fish activity around the restored plots, because as the habitat increase, that attracts some of the fish in. And over the longer term, the team in the Philippines has been able to show that as you increase the numbers of corals, coral cover, and diversity increases, we get increased commercially sized and commercially important species of fish coming into these restored areas. I'll just ask Dexter to now talk to this slide, because this is a great example of where restoration can be effective as long as you can saturate the system with larvae, which used to happen naturally and no longer does on a lot of these degraded reef systems.
Okay, so this is one of our very exciting experiment here in the Philippines in the 100 Island National Park. So from the initial experiments that you saw earlier, we covered the degraded reefs with this mesh matting. So if we really want to go in a larger scale, it's a bit impossible to really cover a whole size or even a hectare of reef. So what we did, as Peter showed earlier, that we have a floating larval ponds. So we collected millions of gametes from natural spawning, and we cultured those gametes in a floating pond close to the degraded reef. So what happened is, after they became competent, we opened the bottom of that culture pond just to naturally release the larvae on this degraded reef without any restriction. So that's around 18 million. So this is how the plot looks like. Totally barren. It's just consolidated rubble. You can't see any corals or some padina or macroalgae only. And this is how they look like after 3 years. So that is how quickly the reef recovered. And after 3 years, these corals started to be reproductively matured. And again, what we do is we always wait for the corals to spawn and use that spawn to restore different degraded reef in the 100 Island National Park. Actually, I'm here on site because it's March and we're waiting for these corals to spawn. And we have a dribble and signs of spawning last night. So hopefully tonight is the night, and we will be collecting again these gametes and will be used to restore part of the 100 Island National Park.
Thanks, Dexter. So this is a great example of near hectare scale larval restoration outcomes. So where are we situated now? Well, we've done some further optimization of the larval restoration process, and I was fortunate enough to receive a grant from the Paul G. Allen Family Foundation for the Great Barrier Reef work. And as a consequence of that, we developed these inflatable larval pools and 5-phase approaches to the larval restoration that we've spoken about. And I think all of you now have access to the report about that and a recent publication on global patterns of coral reproduction, which sort of underpin this talk. As Dexter was saying, we're now scaling this process up in the Philippines. We're working in 6 regions and we've engaged with more than 700 stakeholders. These include local community representatives, local government units, government, university partners, hotels and tourism businesses, a whole range of different important stakeholders in different regions. And the really exciting thing is that after a couple of years of support from our team, some of those local groups are now engaged in their own process and bidding for grants and support to start taking control of the larval restoration process on their degraded reefs. Also, through the Australian government, we've got some further support to expand this process to train our colleagues in Indonesia. As I've mentioned, we've been working extensively on the Great Barrier Reef. So myself and a colleague, Chris Doropoulos from CSIRO, have been heading up a large-scale project called the Moving Corals Project as part of the Reef Restoration and Adaptation Program funded by the Australian government. We've generated hundreds of millions of larvae, and the latest cost modelling on that production process indicates that we're now getting to scale where the average cost of a 1-year-old coral produced from settling larvae is down to about 50 cents Australian. So at that sort of cost, we can start to be thinking about much larger scales of cost-effective restoration. We've also been working with tourism partners in the Boats for Corals project, and we've trained the tourism partners up in this technique in the Central Great Barrier Reef region, and they're now generating hundreds of millions of larvae, each of the main spawning systems each year. We're also working with colleagues in the Maldives, where we're training more than 70 stakeholders from 4 different atoll regions and expanding our stakeholder network with non-government organisations, government department representatives, many reef hotels, and local communities on local community islands. And therefore, we're now at the point where, as part of the WIO Restore project, also funded by CORDAP, we want to focus on capacity building and training with regional stakeholders. And we're going to focus our attention initially in Madagascar in late October, early November. We had planned to try and do this in Kenya, but with the chaos of global travel at the moment, we've had to postpone that. And so initially we're going to be working in Madagascar and then in Kenya early next year. So to summarize, we know that many different reef experiments have shown that increasing larval supply can increase settlement. We've seen rapid coral recovery even on some really badly degraded reef systems with virtually no corals and no nearby spawning corals in the Philippines and in some areas of the Great Barrier Reef. We've been able to re-establish a wide range of breeding populations from branching corals, brain corals, etc. within 2 to 5 years. We now consider that this process can be cost-effective and scalable at larger scales, given the low cost of generating 1-year-old corals. And we're constantly trying to improve the methods, optimizing the technology, and scaling up by scaling out, by building regional partnerships with local community representatives in many different regions. We're breeding from heat-tolerant bleaching survivors, and as a consequence of that, other researchers have found that if you generate larvae from heat-tolerant adults, many of those larvae tend to be heat-tolerant themselves. If we add to that further resilience adaptation potential by looking at scaling up heat-tolerant symbionts to add to these corals, we can then start thinking about some enduring success outcomes to increase the overall resilience of the larval restored populations. And as I've shown in the previous slide, we're now in the process of scaling this process up with local stakeholders throughout many regions of the Great Barrier Reef, in the Philippines, in the Maldives, and now in the Western Indian Ocean. So the take-home message is that larval restoration can enhance recovery even on some badly degraded reef systems. Thanks for listening, and I'll now pass over to Vera.
Thank you for that, Peter and Dex. I could see there's some activity in the chat already about questions, and hands are coming up, Tim, Omar. But I would like to request for some patience, so because we're short on time as well, and we want to make sure we'll get through all of the presentations. So please save those questions, or you can pop them in the chat, and then we'll address them in the moderated Q&A later. And we'll try to, if we don't get to respond to all of the questions, we'll answer them in the email. All right, so I will pass on to Gildas now for the ARMS presentation.
Thank you, Vera. Do you...
Do you want me to do it for you, or do you want to do it on your own?
I can do it. No problem. Let me
Okay.
just quickly make sure. Well, as you are on it now, please do it.
Okay, all right then. Cool.
Yeah, so hello everyone, and thank you for being here so lot. I see 58 people here, which is really amazing. And my presentation will be focused on the autonomous reef monitoring structure and how it will be included and involved in this project. So can you, next slide, please? Yes, thank you. So you all know about the ARMS autonomous reef monitoring structure, which are actually a standardized 3D collectors of marine life used to monitor coral reef biodiversity. So you put these, cube of, assembled plates of PVC under the water, like for from 6 months to 1 year generally, and it can be left more time in the sea, and then you release them from the sea to analyze the biodiversity that are recruited on the arms, and you can get the data are like you can get more than 85% of the reef biodiversity from these arms, and that you analyze either by visual analyzing analysis of biodiversity. In Toliara, can you, next slide please, Vera? So in the southwest, in Madagascar, we have a previous project called Arms Restore. Can you go to the next slide, Vera? Yes, thank you. Yeah, and the project is about to know how to use these arms that are collecting the biodiversity to restore coral reefs, but not only restoring natural coral reefs, but to use them to seed artificial reefs. I will come back to that after, but as plastic standard arms are not available locally in Madagascar. So we ended up adapting it to locally available materials like limestones, for example, because not only it's plastics, but it was expensive, very expensive to import them from abroad to Madagascar, and we wanted to work with communities and work with low-cost materials. So we adapted these arms, and we used limestones available locally, and that the community members can assemble themselves, and we use these limestone arms for our experiments, which consists of next slide, please, Vera. Thank you. So the arms that we are using is not only to census the biodiversity of the on the natural reefs in the region, but we use them to seed artificial reefs. How it works. So we put the arms on a healthy reef in the area for at least 1 year, and then we move these arms with the whole biodiversity recruited into artificial reefs, newly constructed artificial reefs, which are aggregation of limestone stones actually. And the idea is to increase the biodiversity on these new artificial reefs, and not so that the artificial reefs will not only like aggregate fish around and within from the natural reefs, but constructs also a new biodiversity, a bit biodiversity from the arms, which recruit again, including cryptic biodiversity. And this is how we aim at using these materials for our previous project that is called Arms Restore. And currently we are still monitoring the results from the artificial reefs with arms and artificial reefs without arms that are used for control. Next slide, please.
Is it not loading for you, Gildas?
Next slide. Do you hear me?
It's already displayed. Or you want another
okay.
one?
It's late from me. No. It is good. It's my okay. So here, what is important for arms as well is that we really want the involvement of the communities, because what Peter and Dexter just presented earlier, within the context of countries like Madagascar or probably Kenya as well, the involvement of communities are very important, especially that knowing that the reef fisheries is a huge component of the coastal management in the area. So involvement of the communities are important, not only that they will integrate the experiment in their approaches, and so that they will respect the experimental designs and infrastructures that are built, but also involving them will also help the project and help the communities to get sensitized and trained about the importance of the biodiversity in the area. So this is just an overview. I won't explain it in details, but this is an overview of the cycle of the project, how we started from community meetings to agree, so that they agree in installation of the artificial reefs, involvement in arms construction, involvement of in the reef buildings, and then we scientists, we are providing our competency in monitoring, but also complementary data, scientific data on choosing the right sites to install the artificial reefs and the arms. And we are not only monitoring reef ecology parameters actually, but also fisheries and aquaculture and human livelihood. All these to later on, after a few years of monitoring, be able to make sure to link the impacts of artificial reefs and arms or without arms to the development and to the development of socioeconomic in the coastal villages that are studied, but also on the human health that we are also monitoring. And this involvement of community is really important as well, as I said, because they will be, they also, the fisheries practice are also one of the pressures on the coral reefs. So as soon as we are aiming to restore the coral reefs in these villages, it is important to make sure that these pressures and these parameters of community and fisheries are well managed and well considered in the experiments. Next slide, please. Yeah, so this is an example of results of what we observed on the arms after a year and a half in the water on a healthy reef. And as you can see, the arms are dominated by CCA, which is very important in terms of coral reef habitat and the foundation as a foundation of a healthy reef. But you can also observe the important coral recruitment on the arms. So the thing is that on the plastic standard arms, the coral recruitment is near zero from the published data on these plastic arms. The coral recruitment is near zero, while here using limestone provide us data about more than 20 coral recruits, for example, on one single arms, which is really important. And these arms are then moved to the artificial reefs, as you see on the picture, and we can also use them in aside or beside other experiments. Like here, for example, we have added some Mars stars with coral fragments, but for this project, we will be using the arms to monitor the coral larval restoration, but also to use them to seed the cryptic biodiversity with the larval. Next slide, please, Vera. Okay, don't see yet the next slide, but
Could you see that it's already displaying
I
the
see it now.
arm? Okay.
Yeah, so here just an overview of how we initially aimed at using the arms and the larval box together. The idea is first to use the arms to monitor the biodiversity and to monitor the effect of the larval restoration. So we will add arms near the restoration site, near the restoration station. Either it will be direct larval release or using the larval box. And so we have arms near approximately from 50 meters to 100 meters and control monitoring sites outside this area. And next slide, Vera. And this is, we will repeat this schema by using larval box as well, and by using larval, next slide, by using also larval box with the seeded arms, meaning not only using the arms to monitor the biodiversity, but we want also to combine the larval box and the biodiversity recruited with the arms within a restoration site and monitor the impact with the arms and without the arms afterwards. Next slide. Yeah, next slide again.
It's
Yeah,
the
so
last
I will
slide.
end my presentation. Yeah, last slide, yes. I will
Yeah.
end my presentation with this overview of what we are planning. We started to do a few years ago, and how we envision the work that we are doing here in Madagascar with one area. For example, we started restoring coral biodiversity, but the aim is not only to restore coral reef biodiversity, but you also have to restore the fisheries yields, which is an important aspect of the community's socioeconomic livelihoods here in the region. And I want to make sure here that all these activities, all these multidisciplinary and transdisciplinary actions meet together and ensure that there is a synergy within all the programs, so that the whole area, the whole management area will be meeting the main objective, which are actually in term for Madagascar, I'm speaking. Actually, the communities are the main purpose of what the whole work we are doing, how the communities can be involved and can benefit from the biodiversity. And that's why we are actually conserving the biodiversity. But this needs an integrated decision-making framework and a science-based governance that my colleagues will present later on. Thank you very much. I think I will end there. Thank you, Vera.
Thank you so much for that, Gildas. And then thank you for presenting that framework. It's actually very similar to the restoration continuum framework, that pretty much promotes like a portfolio approach to management and conservation, and a portfolio management and integrated approach. So that way you can consider holistically what you're going to, the management interventions that you could do, from threat management to implementing MPAs to applying restoration. So I'm just going to start the governance presentation, and I will try to catch up with the time. So why governance? So I wanted to start this because, yeah, particularly for the evidence base for why governance matters. So I think a lot of the practitioners here also work in MPAs. We know that MPAs have a well-developed management and governance framework, and a lot of lessons are there available and accessible to people to learn from. But there's a lot of gaps, particularly related to coral restoration. So we have the decade of restoration at the moment, and it's ramped up a lot of restoration projects across the globe, and various international organizations are also supporting its use. So while the science and techniques have progressed tremendously, governance of restoration haven't really caught up. So previous reviews on the human and policy dimensions of restoration have shown that there's been very limited studies and knowledge on how people are engaged, how restoration is regulated and managed after the projects end, how the benefits trickle down. So there's also very limited monitoring and reporting. And last year, a major study published in Nature Climate Change, they surveyed around over 300 marine climate intervention practitioners globally, and they found that coral restoration is being implemented in what they call a scientific bubble and institutional void. So again, the science is advancing, but the governance systems are not keeping pace. They found out that there are few projects that had any formal accountability mechanisms in place. There were limited projects that genuinely engaged local communities. There's also pretty much absent, it's often absent, a strategic governance capacity to connect restoration to broader policy frameworks. So coral restoration in this case doesn't only fail because the science is wrong, but it also fails because nobody is really accountable for what comes next. Because normally in practice at the moment, what we've been seeing so far is that governments that are responsible for coral reefs and the communities who live with these reefs, they are not really genuinely engaged in the process. So these lessons, they're global, but they're also not very unique. So we've seen this problems in the Restoration Governance Project in the Philippines. So it's a sister project to that coral restoration project by Peter and Dexter. And I've been hearing similar things as well during the restoration sessions last year's WIOMSA symposium. So I wanted to read the room and do a pulse check, but I will skip that and just focus on the 3 research activities that we have for the governance. So it's a small component, but we're aiming to pack a punch. So the governance research has 3 interconnected activities, moving from a regional breadth to national depth or case studies, and then synthesized guidance. So activity 1 is the regional governance survey. So it will be deployed through different WIO networks to map governance conditions across the WIO countries on 6 dimensions, including decision-making authority, permitting, monitoring, financing, community engagement and inclusion, and adaptive management. So this is why in the registration form, I asked if you would be willing to provide information on restoration governance, because I will get in touch again to seek your participation and input to the research. Activity 2 is the national case studies. Since we're working in Kenya and Madagascar, we will be using the Ocean Governance Accounting Framework, and this can give us the depth to understand how governance arrangements actually work in practice and not just in paper. So Yvonne will explain the OGA methodology in detail. And the third one is the synthesis. So we wanted to create a WIO regional framework, which includes a minimum governance conditions checklist for practitioners and a policy engagement package, which hopefully the Nairobi Convention can take through the COP. So I'll hand over now to Yvonne for the OGA framework. Yvonne?
So thank you very much, Vera, and good morning or good evening to all of you, depending on where you're joining in from. So as you've heard, my name is Yvonne Waweru, and I'm a lawyer by profession, and specifically an ocean governance practitioner as well. So I'll briefly talk about the ocean governance framework, and which is drawn mainly from the ocean accounts framework. So as you can see here, we have okay, I think I can speak on this slide as well. So we are going to study the 2 countries, that's Kenya and Madagascar, and they both have different governance settings. So for Kenya, where I come from, it's more centralized. The marine protected areas are administered by the central government, whereas in Madagascar, we see a more decentralized style of governance through the LMMA's network, which is very vibrant there. That is not to say that local communities are not involved in Kenya, but in terms of what is dominant, Kenya is more centralized than Madagascar. And there we have our leads there, Dr. Karisa and Dr. Gildas from the 2 countries. So the key question here is how centralized MPA governance enables or limits restoration at scale, and whether community-led governance is more beneficial to sustain restoration at ecological scales. I think the lessons from these 2 contexts will be interesting to see how each governance context works in the context, and what are the key lessons that can be drawn from that. So in terms of ocean governance accounts, so basically this draws from ocean accounting, And these are international standards that are used by most countries. And these are used, for example, to report on GDP through the system of national accounts. We also have the SEA framework, which is the system of environmental economic accounting. So ocean governance accounting is drawn from those two, and it's been developed by what we call the Global Ocean Accounts Partnership. So the framework has been adapted from those two internationally recognized standards. So what a governance account is, it's just a record of institutional, regulatory, and management features of the ocean, and it's categorized or is displayed as tables that record ocean decision making from two perspectives. The first one is the spatially explicit context. This one is more focused on space and the jurisdictions under the United Nations Convention on Law of the Sea. And so it maps the different maritime zones as per UNCLOS and the jurisdictions that relate to those zones, and which and the authorities or stakeholders that are mandated or have the authority in relation to those jurisdictions. The second one is on environmental activity accounts, and this is important to record economic activity. And this includes government spending, taxes, and subsidies by different entities that are mandated or are responsible on coral reef restoration. So the focus here will just be on coral reef restorations and how much is allocated in terms of government budgets, taxes, and subsidy. I think with these two approaches, we'll have a better picture on who's responsible, who's has the legal authority over coral reef restoration, and also the monies that have been allocated to coral reef by the different government agencies. Thank you very much. I hand it back over to you, Vera.
Okay, I was gonna have one more slide, but basically, yeah, I think Yvonne has covered all of the methods already from the OGA. And yes, we've caught up on time almost. And I will take over the question and answer session for Dr. Juliet Karisa because she's been having internet issues in Mombasa. So I'll take that now. I know there are a lot of hands up. So I think we'll do a mix of the chat questions and the live questions. So I guess as the first, because I saw it was the first one to come up, I'll call Peter, Dexter, and everyone to, if you can put your cameras on, so to respond to the questions. I think the first question was for Peter and Dexter. How far apart are the control sites from the restoration plots?
Yeah, thanks. It's a really good question. I've looked at the questions online and a really good range of questions. So thank you all for those, and we'll do our best to answer them quickly. The control sites vary depending upon the type of reef we're working on. In some cases, they're tens of meters away from the larval restoration sites. In other experiments, they're more than 100 meters away. In extensive reef systems, we try and separate them. Quite often, if there's a stronger unidirectional current, we'll have the control systems up current of the larval restoration site because we're aware, obviously, of larval dispersal processes. What's quite remarkable across so many of these experiments is that even in control sites at reasonably close proximity, we're only seeing the larval enhancement close to the release sites. We're not seeing significant larval enhancement and spillover effects of many of those larvae in the control plots. The short answer is it varies substantially, but in most cases where we have the control sites at different gradient depths, we're not seeing a major result of that spatial arrangement.
All right. Thank you, Peter. Should I call a live question from Tim? Hi, Tim.
Hello, Vera.
Hi.
How you doing?
Good.
Yeah, Tim here. Kind of, I'm kind of the reef elder here, so I thought I'd give some advice. One of the things that I've always found interesting about this region is we don't have big mass spawning events.
Mm.
They tend to be very small and kind of scattered. So when I read about these things in Australia or the Philippines or Caribbean, I kind of wonder what's going on here in terms of the timing and the scale of things. We do have breeding going on, and by the way, now is the month when it starts. So, I'm concerned that that kind of scooping up lots of larvae is going to be the bottleneck here. So we have to think about that carefully as to how we can avoid that not being a bottleneck, because I would love this project to succeed. And I'm worried, as a person who runs into problems a lot, that this could be one of the bigger problems. So I'm sort of bringing that to your attention. I'm sure you know there are other ways to do it. You could focus on colonies instead of, the surface water. But a lot of it is going to be finding the right time and place. You could spend a lot of time trying to figure that out, and then even not succeeding. In Kenya in particular, we've lost acropora on a massive scale, and much of that started before, even before I was working here. I started working here, by the way, in the 1979, and I think a lot of corals bleached in 83 and maybe even before I even was here. And then many of our reef edges they have little colonies of, acropora here and there. They can be hundreds of meters apart, some of them actually. So the idea that it could be spawning and producing larval slicks or egg slicks or whatever you call them. I haven't seen that in almost ever. So I don't say that to be critical. I say that to be helpful. That we need to think about this now. I think Tanzania might be a little different, Madagascar possibly a little different, but at least in Kenya, where I spend more time, we need to think about this carefully. Otherwise, we could spend a lot of money and not get where we're going. So I just want to put that out there because everything that I saw was impressive. And again, I hope it works out, but I'm just very concerned about what I would see as the main bottleneck. And some effort to get out even now. Right now is March when our corals are spawning, and see where these corals are spawning, if at all. It might save you some time instead of just showing up as a crowd at one time and assuming it will work out. And you may spend a lot of time in your hotel wondering where these corals are and where the slicks are. Coral cover in Kenya is like 10% now, and a lot of it is the favids. And even those are not doing well. Let me put my hand down, and thanks so much. Yeah.
Thank you,
Thanks,
Tim.
Tim.
Peter, quick
Yeah.
response?
No, they're great and important insights into the local area. I guess in terms of context, obviously we could only cover some of the bigger picture things. So not all of the areas we work in have mass spawning. Probably the most complicated asynchronous spawning place that we know of on Earth are the Maldives, where parts of population dribble spawn over multiple lunar phases. So predicting when these are spawning, but we're still able to find pulses of spawn by using local knowledge, working with local stakeholders. And so there's a couple of ways around that dispersed spawning process. One of them is to create breeding hubs. So if you know that corals have ripe eggs and sperm, you can either temporarily or more permanently move some of those colonies together. So your efficiency of collecting the spawn is increased. We also use spawn cones if we're trying to use selective spawning, and from putting down 30 or 60 spawn cones, which you drop over the breeding colonies in the evening waiting for spawning. It's a relatively simple, low cost and effective way of generating many millions of larvae. And we've done that even on the Great Barrier Reef where we've had dispersed spawning processes. So there are many ways around that. We're certainly not naive. We know from Sangita's early work and other people in Kenya that there are dispersed spawning periods across different populations. We also don't only stick to the irregular spawning acroporid group. We're doing a lot of work with massive corals. And brain corals have a more predictable spawning outcome in most locations. And once you lock into that, it's much more feasible to be focusing on those. So absolutely thank you for those insights. And we definitely take them as in the way they're intended as advice. Thank you. As I said, we had been planning to come over to Kenya and start this process of looking for gravid corals and starting to better articulate spawning patterns locally. But unfortunately, the global chaos at the moment with travel just means it's impossible, which is why we're reverting back to Madagascar for the first thing. But we definitely take those important insights on board. And yeah, please keep in touch and we'll try and take advantage of any local knowledge we have of anyone who's seen either spawning or at least some of that reproductive material in some of these local corals. Thanks, Tim.
You're welcome.
Thanks,
I guess
Peter.
I didn't know the word
Yeah.
dribble spawning, so thankfully you. That's a nice term.
Yeah, it's a new one. We coined it in the Maldives after many
Dribbling.
frustrating nights. They have dribbles, and Dexter's got that in the Philippines right now, where just a few of the colonies are dribble spawning, and it's very frustrating. But it's a matter of knowing how to deal with that process and wait for some more synchronous spawning, or use spawn cones to take advantage of anything that is spawning.
Yeah. Okay. Anyway, it's age appropriate dribble spawning, and Kenya
Yes.
appropriate.
Yep.
So let's hope it works out. I just would recommend probably if you can get people out in the field pretty soon to
Yeah.
sort of do some surveys because this is the month.
Yeah.
We're having a very unusual time of weather here, by the way. Our rains have come 2 months early.
Okay.
Yeah.
Makes it more complicated.
Yeah, so I'm even begin wonder what's going to be going on with the spawning. Anyways, anyway, best of luck in your
Thank you. But
your
please keep
project.
in touch. We value that information. Thank you.
You're welcome.
Thank you, Tim. We'll go to again one question from the chat. What's the criteria to select the degraded reef for the larval restoration?
Dex, do you want to?
Peter or Dex? Yeah.
Yep.
Oh, yeah. So, yeah, there's a criteria in really choosing the right degraded reef. So, sounds weird, but it's actually very important because that will dictate the success of your larval enhancement intervention. So, if you choose the proper degraded reef. So, for us, it seems like the number one criteria is, of course, it should be a barren, hard substrate reef, not covered with macroalgae. So that's very important, and not covered with sediments. So unfortunately, our sites here in the Philippines, as you saw earlier from Peter's presentation, it's a rubber field, but it's consolidated. So it's important that we have a consolidated rubber field if you want to restore a rubble field area, because I saw one question in the chat box. Because if you will put the larvae there, they might settle, but of course, they might move and die eventually. So basically, because the larvae are settling on the substrate, so it should not be covered with sediment or macroalgae. So yeah. And Peter?
Yep. Thanks, Dex. Yeah, so we have had some success with some algal dominated reefs, but the growth rates are often reduced. And so, yeah, thinking carefully about trying to find sites that are lacking corals that haven't yet been smothered by soft corals, macroalgae as competitive organisms is often very important in terms of the choice of site.
All right. Thank you. I'll ask a live question. It's from Madagascar. I'm sorry, Ran Gianarino.
Yes.
Yes, thank you for this webinar. Hi, everyone. So I have a question. Maybe I relied on the last question on the chat. Sometimes, larval supply is not the limiting factor on a degraded reef. Probably we can have a lack of suitable substrate for settlement. So I'm wondering if whether there are prerequisites that should be made before releasing larvae, or for example, you have a plan to deal with this to maximize the effectiveness of such techniques.
Yeah, thank you. Both of those questions are really good. and they correspond. So we're answering two questions at once, which is very time efficient, which is great. Look, no, those questions are really important because we know that unstable rubble reefs are not good substrate, even though they may have lots of coralline algae. Sometimes the next storm mobilizes them, so any of the naturally settling corals, can be killed by turnover and things like that. One of the really interesting things from the Philippines work that I showed and Dexter spoke to was that was a highly degraded rubble reef. And theoretically, it's not a site that you should normally be thinking about doing larval restoration. I think one of the key learnings from that is that because the team swamped that system with larvae and they had such extensive settlement, as soon as the corals started spreading their basal plates over the reef system, they started locking in that unstable rubble. It was also fairly old rubble, so it had been mixed and stabilized by storms, etc. But what we have seen is that, there was, in the early years of that, there was more than 100% coral cover. So overlapping tiered plates of brain corals and plating acropora. So the outcome of that clearly shows that on some rubble reefs, as long as you mass supply larvae, which happens naturally in pulse supply systems, you can overcome the limitations of the rubble system. So you just have to be careful. If it's a system that's going to be unstable, you probably do need to think about either going to another site, or if that site is incredibly important for some particular reason, you would have to think about rubble stabilization. And there's a series of techniques that other restoration people have been using. But in general, it's, again, thinking carefully about site selection and the extent to which a rubble reef is likely to be mobilized by big storm events.
Right. Thank you so much for that, Peter. I'll ask, there's a question in the chat and I can answer this maybe from a governance and management perspective. Catherine asked, integrated reefs, particularly those dominated by rubble or turf are, no, not that one, sorry. Sorry, it was Joanne's Kawakar's question. What is your advice to ensure successful reef-based larval propagation in areas where fishing is taking place? Is it a matter of survival for the larvae, or extra measures such as closed areas will still have to be considered? So I wanted to answer this question because, we published a paper last January in Restoration Ecology regarding management and governance framework for restoration, and particularly integrating it into the Philippine context. And this basically synthesizes some of the lessons that we've learned in the Philippines because, restoration requires a certain site selection criteria. From a governance perspective, we found that, local governments that have more experience in marine conservation and also coastal resource management are better positioned to sustain the larval restoration and manage the restored reefs. We've also currently working on another paper documenting the lessons that we've learned because we've been preparing we've basically included development of management plans to be able to protect those or manage activities in the restored reef areas that Dexter have worked on. So those are the recommendations, and also in terms of site selection criteria, there's also some requirements in terms of threat management, since water quality is a pretty important issue, among other things. So I think that's that answer. I'll ask another live question and then another chat question, and then we'll wrap up. If I don't get to call you, please type your question in the chat and we'll try to answer them via email. Okay, Isabel De Silva.
Hi, good morning. I'm from Mozambique, and it's a little bit of a follow of Tima Clam. So I've been following probably the only big mass spawning of the Western Indian Ocean in the north of Mozambique in Vamizi Island. I'm following since 2006. It's in between three islands, like 200 kilometers of coast that I've been following. So Over there, the season is September, October for Acropora mass spawning. So for me, it will be very interesting to collaborate, and probably I have funds from another coral restoration of rubble beds with spiders that I can drag some of that money for traveling for the sites you will be doing. So I would love to learn this way of using the coral spawn of the north around. So just if you want, I can send the papers from all the information about that coral spawning. And connecting with other talks about being very split spawning. During these 20 years that I follow every year the spawning, I found that in the last 6-7 years the spawning was not in one day like before. The first 10 years was always in the same day or same 2-3 days. And I saw a lot of splitting between 2 moons, the spawning, and the slicks appearing smaller and in 2 different moon cycles. And I think was very related with the climate change and the change from the dry season to the rain season changing and winds changing and appearing in the wrong time that changed the events of mass spawning. So it was not a question, it was more about speaking about my experience with spawning and my willing to participate, even if I don't belong to the process, but if I could get some of my team to participate in some of the activities, will be very good. Thank you very much.
Thank you, Isabel. Before I give it to Peter, there's actually a similar question on the chat that Jennifer O'Leary asked. Can groups outside the target countries join in the training? Peter, do you want to respond to this?
Yeah, look, thanks, Isabel. That's really important information. And yes, let's connect and try to see how we can enable some teams. So at the moment, we don't have project funding to enable people from outside the main groups and the main nations in Madagascar and Kenya to travel. But if there are independent funds available, then yeah, we will definitely encourage and try and enable as much as possible to take advantage of the CORDAP funding to increase the training and capacity building we can do together on site. So some of the work we do can be done online with some more detailed presentations, which we plan to do. But nothing beats being on the reef and doing the process together and chatting and learning about it. So wherever we can do and wherever it's feasible with some additional funding from local groups, we would definitely be thrilled to have some other people involved. So thank you, Isabel and others. We'll obviously have to try and manage that process in terms of numbers, because we usually end up with greater numbers of people really wanting to be involved than we have capacity for. But we'll definitely encourage from people. This isn't designed to be a closed door system. It's designed to be as open as possible to see how widely we can support other processes within the limits of our funding and other availability.
There was a follow-up regarding online training, and maybe you could talk about what we were planning for the training.
Yeah, so the online training will consist of a series of sort of more detailed presentations about each aspect of the process. So as we've already presented briefly here, there's the different phases of the larval restoration. There's the ARM system that Gildas presented on. Vera and Yvonne are looking at some of the governance processes and applying different models to that, and we've got some really fascinating differences between Madagascar and Kenya in terms of whether or not it's centralized or decentralized in terms of management. So all of those processes will have a series of online training for that we plan to do. We're going to be setting up a series of stakeholder conversations, processes as part of this. And then wherever possible, we'll try and enable some on-reef training as well during the spawning periods.
All right. I think Peter also started covering the next steps. So it's a good thing for those of you that said that, yes, we're happy to receive updates. We will definitely give updates. And also for those that have said, yes, they wanted to provide information and share information regarding restoration governance in the WIO, we will also get in touch with you guys. So I don't think I need to cover a lot of the next steps, except for the near future is that, yeah, we'll try to answer all of the remaining questions in the chat. And before we close this, if you still have any burning questions, you can type them there. But then I guess that's it for us. Thank you so much for attending this Inception meeting. I can give it back again to our project leader, Dr. Arthur Tuda, to formally close this meeting.
Right, great, Vera, and all the presenters and participants. Thank you very much for finding time to join this session. This is an introductory part of this project, and we will really wish to keep engaging with all of you. So feel free to write to us, WIOMSA, and the rest of the project team. We would wish to, as Peter said, involve the whole WIO in this learning process. It's basically building a strong network of practitioners, managers in the region, and we'd like to see that as one of the key outcomes of this project. So those who have projects that are running that are similar, please let us know, and we see how best we can partner and work together on this aspect. So thank you very much, Vera. I don't have much to say, but to wish everyone a good week ahead, and see you soon. And please keep in touch. Thank you.
Thank you so much. I'm sorry I went over time, but thank you so much.
Thank you,
Thank you,
everyone.
everyone. Bye.
Okay,
Thank
bye
you.
now.